I've been taking a long time to make this video, partly because of things going on at work, the start of a new project and my recent promotion, but also because I wanted to get this video just right. In my own estimate, if we let the biology alone determine these divisions, there are either about 350 human races, about 250 of which reside exclusively in Africa, or there is one human race.
Your assertion is not supported by your own figures.
When genotype, someone who self-identifies as African American will typically have between 5 and 20% European ancestral markers. The same for those who identify as European Americans. Most will be unaware of very recent African ancestry.
No whites are even represented on your plot. As you can see in the attached figure, there is virtually no overlap.
This is called admixture, the mixing of different ancestral populations in the genetic complement of a modern population through interbreeding.
Your caption states that European Americans average 5% African ancestry, while the figure shows 3%.
There are no half-robin, half-sparrows. That's simply not how taxonomy works.
You know, Kirk Cameron could sue you for plagiarizing his crocodile analogy. Only his analogy makes a little bit more sense. All subspecies within any species can, by definition, hybridize. If they can't, then they are not subspecies, but full species. Many full species can also hybridize and generate fully viable offspring. What you label as Asian and European is actually within versus between populations, respectively. There are both Asians and Europeans on both distributions. Also, the purple area does not represent misclassified individuals. Only half of them would be misclassified. And the overlap between Europeans and Asians is not a third, it's 0 when enough markers are used.
But I want to clarify early on the idea that an alien coming to Earth, with our same understanding of phylogenetics, would put all modern humans into a single species and a single subspecies. We are all Homo sapiens sapiens. Any differentiation from this point on will have to be below the level of subspecies.
Not according to your own figure, in which humans are labeled Homo sapiens, not Homo sapiens sapiens.
In classical taxonomy, a Deem or Klein would fall below the level of a subspecies.
Wrong. Kleins and subspecies are not mutually exclusive. Range species, for example, are Kleins that are above rather than below the level of subspecies.
So how do we define whether a group is a subspecies, race, Deem, or Klein? The famous geneticist Sewell Wright, the man who wrote most of the equations that we use today in the field of population genetics, developed a particular descriptive number called the FST, or Population Fixation Index. It was a way of describing something we call population substructure, that is, whether a large group is genetically homogeneous.
These FST values are all false. According to the latest literature, FST in these three species is roughly 15 to 20%, which is about the same as for human races. Of all these species, only Grant's gazelle has a slightly higher FST of 20%, and perhaps even higher since FST tends to be underestimated in microsatellites. So let's say they have a 30 or 40% FST. But this is not just a random species. Look at the title of the paper from which your data originated. Not that you actually read the title, let alone the paper itself. The title is Extreme Genetic Differences Among Populations of Gazella grantii, Grant's gazelle in Kenya. According to the abstract of the paper, these differences are on the same order as between distinct species of gazelle.
The FST for modern humans is approximately 0.110, or we could say 11%. What that means, in essence, is that 89% of all the variation in humans is shared across all groups. Only 11% of human genetic diversity can best be explained by the presence of distinctive subgroups. Wright himself proposed that a subspecies should be considered valid when this FST value exceeded 0.25. Humans don't even make it halfway to the standard criteria, so objectively, by the standard taxonomic practices governing subspecies, human populations don't qualify as subspecies.
No, Wright never proposed such a thing. And you know why? Because he wasn't a genetic determinist. Only a true genetic determinist would determine the taxonomic rank of demes based solely on genetic differentiation.
Each dot on the PCA graph represents a group of individuals drawn from a different population.
Actually, no. Each dot represents a single individual, not a group.
The goal here is to see how much of the variation in the dots can be accounted for by our two marker sets.
What two marker sets? I only see one. I think you mean two sample sets. The markers are the same.
If human races are distinctive demes, this is what they would look like. However, what happens when we sample from all those geographic regions in between these distant populations? Now we can see what looks like a continuous change from one region to another.
What you labeled as Africa and Europe is most certainly not Africa and Europe. Just look at the distances on the bottom of the plot. European and African populations are not 2,000 kilometers apart. It's more like 6,000 kilometers, which, if anything, would imply that the genetic discontinuity is between Africa and Europe. All this plot shows is that distant populations are more genetically distinct. Now, what exactly did you expect? That distant populations would be less distinct? This trivial observation would be expected within virtually any species, except perhaps among island subspecies. The white-tailed deer, which you claim has distinct subspecies, shows a similar pattern. And note that the distances between white-tailed subspecies are actually lower than expected given the distances within some subspecies. So by this standard, humans are more differentiated than white-tailed deer.
For example, geneticists can map a cline of Spanish markers into Western France, or differentiate French and German-speaking Swiss people. To me, this suggests that dividing up diversity into homotypic groups is probably doomed from the beginning.
Nobody uses the term homotypic in the context of human races except you, Concordance, and a few other anti-race crusaders. And FYI, the proper term is monotypic, not homotypic.
Let's take our PCA graph and try some other arbitrary divisions. What we're going to alter here are called inferred populations, and we assign the capital letter K to this number. Set K equals 4, and we have the basic continental populations. Again, the data isn't changing, only how many inferred populations we assume. If we increase K to 9 inferred groups, you get an analysis that still looks quite valid. There's nothing in the data here that tells us what value K should be, unless we already have a goal in mind.
The human analyst tells the program how many clusters or groupings to create. Recall that this arbitrary and inferred number of groupings is called K.
Wrong. The inferred number of clusters is given by the program, not the human analyst.
So how does my simulated data compare to actual PCA analysis of human populations?
I rearranged your plot and superimposed it on a map of the world. The Italians plot with the American whites over the British Isles. The Chinese and Vietnamese cluster with the Japanese over Japan, and the Sub-Saharan Africans cluster over Antarctica. Feel free to pause the video and see for yourself. The dreaded clines you keep mentioning are common not only among subspecies, but often even among distinct species, such as coyotes and wolves. Canine phylogenetics are much more messed up than human phylogenetics, with some wolves like the red wolf clustering with coyotes. So let's abolish canine taxonomic rankings altogether, said no taxonomist ever.
Humans are one of those rare species that span the globe without geographic isolation.
The white-tailed deer, which you named as having valid subdivisions, is also quite mobile, reaching speeds of up to 75 kilometers per hour, and with no obvious geographic barrier between most subspecies. And yet, there are currently 38 recognized subspecies of this particular deer, including 14 in Mexico alone.
We are all mutt blends. We have reason to believe that there was some intermixing between anatomically modern humans and the Neanderthals, possibly other ancient hominids. We can set an upper bound to this contribution at about 3% of markers. There are some pretty silly ideas that Europeans somehow contain genes from Neanderthals that Africans do not possess. This is simply not the case. At most, there are some variations in certain groups of modern humans that are best explained by admixture with these ancient hominids.
So 3% admixture within a species makes us all mutt blends, but 3% admixture with a different species is meaningless and silly.
James Watson, who received the Nobel for his work in discovering the structure of DNA, in a newspaper interview, made comments that linked the social problems in Africa to the lower genetic potential of Africans. It was an off-the-cuff comment, the kind of thing that pops up in casual chat with someone who grew up in a different era. The controversy he sparked led to his resignation as head of Cold Spring Harbor Labs in 2007.
In other words, if you dare to challenge the quote-unquote scientific consensus on race, you'll be thrown under the bus regardless of your scientific credentials and accomplishments.
James Watson was one of the first people to have his genome sequenced, so his genetic markers are publicly available on the internet. The Icelandic genomics company, Decode, was reported as claiming that the Watson genome showed greater than 15% African ancestry. The data is still somewhat questionable, since the early genomes were very poorly annotated and aligned, but this would not be out of the ordinary.
I agree that Watson's African ancestry probably isn't out of the ordinary for a white American, not because many whites have 16% cryptic African ancestry, but because I don't believe Watson has anything near 16% cryptic African ancestry.
It also shows that no matter how European someone appears, you can't imagine that they are by any measure of pure ancestry.
Children, and especially toddlers, have lighter skin, eye, and hair color, and are generally harder to classify. Nevertheless, I believe I could still classify these children.
Notice, for example, in this global population, that none of the bars is entirely of a single color when we allow for a larger number of inferred groups, that is, high value of K. Every tribe or ethnicity contains within its gene pool plenty of mixtures of markers from adjoining groups.
And now let's see a similar plot for North American wolves, which you previously cited as an example of extreme differentiation.
If we zoom in on just a few genetic variations that happen to be very different between, in this example, the Yoruba people of Africa, the French people of Europe, and the Han people of Asia.
Wrong. It's not French, it's European Americans. And it's not Han, it's a combination of Han Chinese and non-Han Japanese.
There are about 3 million of a particular class of genetic variations called SNPs, single nucleotide polymorphisms. But a lot of them are only measured in a few individuals in the whole world, so we should consider the total variation as about 900,000, which I'll round to 1 million for this narrative.
Wrong. There are over 100 million SNPs, and if we include rare and private variants, it's closer to 3 billion than 3 million.
There were no genes associated with athletic performance or brain development that were identified as being under positive selection in a single group.
Wrong. At least one of the genes in your figure has been recently associated with brain function and development.
Thus, if we were to generate a proper family tree of humanity, and if it was to represent all of human diversity, it would have to be rooted in Africa. Out of every 10 branches, a good 7 or 8 would still reside in Africa. Europeans, Asians, Australians, and the first Americans might all be twigs off a single branch. We might call the branches, for example, Pygmy, San, Yoruba, Bantu, and then all the non-Africans would be lumped into Amero-Eurasian, with Australian Aborigines possibly being their own branch.
The Bantu and Yoruba are not two distant African branches. They're actually quite close. If I superimpose an MDS plot on the globe, where the first dimension of genetic distance is calibrated on the geographic distance between South African Bantu and Nigerian Yoruba, Europeans will cluster on the moon. As for the more distant African branches, they aren't as distant as you might think. The roots are distant, but there has been extensive gene flow since then. Not so much between Africa and Eurasia, as you claim, but among African populations. So although their roots are far apart, the actual DNA is closer.
What we really are talking about with race are very, very extended families that blend together at the edges. You may notice similar characteristics in your own extended family, a certain type of nose or eyebrows, or in my own case, abnormally large craniums run in my family, at least on my father's side.
For someone that casually dismisses any correlation between brain size and intelligence, you seem quite eager to plug your supposedly oversized brain.
But it means we approach each of these cases with a high degree of skepticism. This isn't about political correctness. It's about our ability to fool ourselves, our tendency to fall victim to our own confirmation bias.
Every cell of each plant and animal contains genetic information coded onto the DNA molecule. The model